So if you want to do a multiplex experiment, this means actually that you will target many different genes at the same time in one experiment. Sometimes a gene can occur in different forms, which we call alleles. And it's also possible, of course, to just target all these different variants of that particular gene. And this is then called a multi-allele experiment. So when you design a multiplex experiment, you have to take into account, again, that not every guide will be as efficient or top performing. So for example, if you want to target 6 genes at the same time using one guide, it can be very possible that in, let's say, you have 20 transformants, that in all 20 you have the same gene mutated, but maybe your 6th gene only in half of them, or maybe in one single plant only. Yeah, so if you make a vector, or if you want to target 6 genes at the same time, you again have to take into account that the guide RNAs can be variable, so not all of them will be as efficient. It is possible that one gene will be mutated in all of your samples, so with your top performing guide RNA, but maybe with your lesser performing guide RNA, only in 5% of your plants you will see mutations. So if you can mutate a single gene, multiplexing is usually the obvious next step. And then again, you have to see how far do you want to take this. Do you just want to mutate 2 genes? Do you want to mutate 18 genes? So it depends, of course. If you want to edit a lot of genes at the same time, of course, your chances that all these genes will be mutated in your plants will become very low. So, but if you only want to target 2 genes, of course, your chances will increase.
When you do a multiplex experiment, you also need to do a multiplex PCR. So instead of only doing one PCR for one target, you need to do, for example, six PCRs all at the same time. This can sometimes also be an issue with efficiency. Amplifying one gene is often more efficient than doing amplifying 6 genes at the same time.